Well, thank you everybody for coming today. I know things have been a little crazy with the new system and doing things online, so I appreciate everyone tuning in to listen to me talk about some data today. I'm pretty sure I've met everybody at least once, if not more, but just in case I haven't, my name is Lindsay Milich. I am I guess fifth year neuroscience PhD student working here at the Miami Project with Jay Lee. So our lab primarily focuses on mechanisms of wound healing after contusive spinal cord injury. So that's really what I'm going to talk about today. I'm going to present some of the work I'm doing for my thesis, which focuses on the innate immune response, or the myeloid immune response to spinal cord injury. I'm primarily looking at this using a single cell transcriptomic analysis. So I'm gonna get into a little background. I'm sure that everyone is a little bit familiar with spinal cord injury pathology, but like I said, something that we're majorly interested in is looking at the different wound healing mechanisms that occur after contusive injury when we use a mouse model. So there are several different pathologies that we're interested in, but the immune response to injuries is a very big one that we've classically studied over the years, and that's really what my thesis focuses on. So I'm really going to look at the myeloid immune response to injury and how this can influence other pathologies to progress. So the other two pathologies that I'm also interested in looking at is the vascular remodeling. So there's a lot of vascular disruption after acute injury, and then a revascularization event that will occur after starting a couple days after. So this is something that I'm interested in looking at, specifically how the immune system can modulate this response, and also the scarring response, the two different scarring responses that occur after injury. So the glial scarring response, and then the fibrosis that occurs in the kind of lesion epicenter. And I'll explain more of why we're interested in looking at the connections between these different pathologies a little bit later in a few slides. But the majority of my talk is going to be focused more on the immune response and how figuring out how this can be influencing regeneration after injury. So a couple, before we get into kind of the guiding questions that I'm interested in, I mentioned very briefly that the major assay that we're using for this study is single cell RNA sequencing. So this has been around for a little bit now, and many of you have probably heard about it or know a little bit about how it works. But basically, we wanted to take this route over a traditional bulk RNA sequencing assay because you get transcriptomic analysis at an individual cell level. And this is really important for the questions that we're interested in answering, because there's two major advantages that specifically for my project that is I think are very essential. First, you get, like I said, a transcriptomic profile of every individual cell in the tissue sample that you submit. So this really allows us to look at the transcriptional heterogeneity that exists within the myeloid population itself. So one of the major questions I'm looking into answering is how do we break down these myeloid populations, and do the different cells act differently after injury and different time points after injury. And another thing that is really important for this study is looking at how the myeloid cells interact with the other cells in the nervous system and how they influence pathology. So we need to be able to identify each individual cell and what they express, to see how they might be interacting. So this is a very essential platform in order to answer some of these questions. Just very, very briefly, why this is unique and how this works exactly. So instead of the PCR reaction occurring in the entire cell sample, uses the power of an oil emulsion droplet to capture each individual cell with all of the components it needs for the PCR reaction. And it's able to be barcoded or tagged in that way, and then all of the transcriptomic information can be bioinformatically tracked back to each individual cell. So this is very exciting for us. It opens a lot of new avenues, and we can answer a lot of new questions that we weren't able to ask before. So moving into a little bit of the agenda of what I'm going to talk about and kind of the guiding questions for my study. This kind of, it operates in two parts. So firstly, I did briefly mention this, but I'm interested in the heterogeneity in the transcriptome that exists within the myeloid populations. So not just a macrophage or a microglia, but maybe subpopulations of these cells that could be differentially influencing the pathological progression after injury. So we have a lot more granularity in what types of questions we can ask here. And then the second part of this, looking outside of just the myeloid population, how can this immune population be influencing or contacting the other cells in the injury site and influencing the progression of these other pathologies that affect regeneration. And I'll explain a little bit of how we went about analyzing the interaction between these cells when I get to that. But first, I'm going to start with looking at the myeloid population itself and how we did this, and look at some of the subpopulations that we're able to identify within the broader myeloid populations that we all know in that exist in the spinal cord. So this is a very simple kind of overview of the methods here. We used C57 black 6 mice. They were given a 65 kilodyne spinal cord injury. And then at various time points after injury, an 8mm section of the spinal cord surrounding the injury site was collected, enzymatically processed, and then submitted for this sequencing. So we used the 10x genomics sequencing platform. This is, I think, one of the most popular ones out there right now, and we've been very happy with it. So if anybody's interested in doing this, I would suggest maybe this platform, and I explained how this works exactly. So I wanted to mention all of the bioinformatics was done by James Cho in our lab. He's a technician, so this would not have been possible without his help. So we looked at 4 different time points in the acute injury space. We had an uninjured spinal cord for a control, and then we looked at 1, 3, and 7 days after injury. So this was very important for us to compare how these subpopulations can be dynamically changing over time. And we do have two animals per time point here. So 2 spinal cords were sequenced for each individual time point. So this is kind of the output of what you would get from a single cell sequencing study, just kind of the very beginning. So this is a UMAP plot, which demonstrates, it's a dimensionality reduction technique where we can look at each individual cell and how they cluster together on this platform. So these, each individual dot represents an individual cell. And these populations here were manually annotated by us based on some of the canonical markers that exist in the literature. So we did sequence the entire spinal cord section. So we got all of the cells in the spinal cord, not just the immune cell population, which we were very, very excited about because we're interested in these reactions. So we have the myeloid populations over here, where our microglia, the monocyte populations, the peripheral macrophages coming in, a very small amount of lymphocytes and neutrophils and dendritic cells. We have the vascular associated cells here. So we have a lot of endothelial cells, a few pericytes, and then a fibroblast population, and then more of our CNS intrinsic cells down here. So we have our astrocytes, very few neurons, which is what we kind of expected because traditionally neurons don't really come out very well in the enzymatic dissociation that we use. So if you're interested in neurons, I would suggest more of a single nuclei approach instead of a single cell approach. But we also got the oligodendrocyte and the oligodendrocyte progenitor cell populations. So this is very exciting. This combines all of the cells from all of the time points. So this is the uninjured 1, 3, and 7 day time points from all of the animals. So now that we have these populations, we can start to ask questions about the actual transcriptomes of each individual cell here. So kind of going back to my first guiding question, what is the actual transcriptional heterogeneity that may exist within the more broad myeloid populations that we see in the acute injury phase? So in order to do this, we basically isolate the myeloid populations we're interested in. So this includes both the peripheral and resident microglia populations, so the macrophages, monocytes, and the microglia. We isolate these populations bioinformatically and then perform a clustering analysis over again to isolate the heterogeneity only within these populations. So when we do this, we get another UMAP output with the populations. So I'm going to go through the peripheral first and then the resident microglia subpopulations next. So this is a lot. So just to orient everybody, this is another UMAP plot here, the same as the setup as the last one, but these are just including the myeloid populations we isolated. First, I'm going to show the peripheral myeloid populations here. So this is the UMAP, and then these are the marker genes that we use to identify these cells moving forward. So very briefly, we have, and again, these were annotated manually based on the genes that they expressed. So we have monocyte populations here that we were actually able to break up into two subpopulations of monocytes. So there's a smaller one here and a larger one that exists here that we're differentiating based on their, majorly based on their expression of Ly6C. So we have a Ly6C high monocyte population and a Ly6C low. We did get the neutrophils again as we expected. And then we have a couple different mature macrophage populations that came out of this as well. So I want, first off, this BA or border associated macrophage population, which is makes up a smaller portion of the total cell population here, but this is basically macrophages that are traditionally located at the border of the CNS and the peripheral nervous system. So, and these have been described a little bit in the literature before. So that was interesting that we were able to annotate them based on what they express. And then we have these other two mature macrophage populations that we termed A and B. So these make up the majority of the mature macrophage population. So B and then A is right here. And then I'll go into a little bit about what makes these two different in some of the coming slides. So this is the peripheral myeloid populations that we were able to, subpopulations that we were able to identify. And then in the same analysis, we were able to look at the resident myeloid or the microglia. So this is the same here as the marker genes that we use to kind of identify these going forward. And we have several subpopulations of the microglia that we see over time. So again, this is, the UMAP is a combination of all the different time points together. And this allows us to track how the proportional contribution of these cells to each time point over time. So we have a microglia population that we termed as the homeostatic, which were fairly prevalent in the uninjured population. And then we get into several subpopulations of microglia that appear after injury. So we termed this DAM, so disease-associated microglia, which have actually been described in other mouse models of not necessarily injury, but disease. So I know that they are fairly well described in Alzheimer's models of Alzheimer's mouse models, and I think also human tissue. They have similar transcriptional profiles. So these are called disease-associated microglia, and we found several of the marker genes that they expressed in our study were also expressed in the literature. So this is why we're calling them disease-associated, but we broke them up in between A, B, C, and D. So the DAM-A population is down here, and then we have B, C, and D, which do have transcriptional differences that I will go into in the coming slides. And then we did find a big population of microglia that showed a lot of cell cycle genes that we termed as dividing microglia, so DIV. So this was very interesting, and this is what the UMAP looks like together. So these are all of the subpopulations of myeloid cells that we found in combination of all of the time points. So I want to kind of highlight the temporal progression of these cells over time, because I think that's really interesting for understanding how they contribute to each phase of injury and how maybe they are differentiating within themselves. So first, looking at the peripheral myeloid populations here. Your faces are over the plot, so I need to move that. So we didn't plot here the uninjured population because the blood-brain barrier is intact. There are no peripherally derived macrophages in the injury site, which is what we would expect. So there's really no data to show there for the peripheral population. So we start the plotting here at 1 day. So 1 day or 24 hours after injury, we have, as we would expect, a lot of the monocyte and these transitioning monocytes. So the monocytes that are kind of the Ly6C that are later in the progression here. So by the time we get to 1 day, a lot of the population is really dominated by the monocyte the population overall. And then by 3 days after injury, we really lose that monocyte population and get into macrophages that are starting to exhibit the mature state. So a lot of these are the macrophage A population, and a little bit of the macrophage B is starting to appear. And then by 7 days, the macrophage A population decreases, and the majority of the peripheral myeloid cells are dominated by this macrophage B. So some of the populations, like the neutrophils and the dendritic cells, they really don't drastically change over time, but we did plot them here anyway, so you can see their proportional contribution. And then when we look at the resident or microglia, we see something interesting as well. So as we would expect in the uninjured condition, the majority of microglia, if not all the microglia, are in the homeostatic state here. And then by 1 day after injury, the homeostatic population disappears, and they are actually all characterized by this DAM-A population, which we thought was very interesting. So moving forward into the 3 day, we have this DAM-B population that appears, a very small population of the DAM-C and D, but we have the majority of the dividing microglia population shows up here at 3 days. And then by 7 days, the DAM-C population is almost evenly split with the DAM-D, and the dividing microglia population goes down significantly. So this is important for us to be able to track their progression over time and see maybe which population is really contributing to pathology at an individual stage. So now I'm going to get into a little bit about what makes these populations different. So this is the peripheral myeloid population here. So these are all the macrophages and neutrophils and dendritic cells. So this is a differential gene expression analysis of just the peripheral myeloid population. And we plotted the top 10 differentially expressed genes in each population, so we can kind of see maybe how they differ from each other and what makes them unique. So there's a lot to take in here. So I'll kind of highlight what we thought was most important to take away. So first, looking at the monocyte populations, we can see here a big difference between the two was actually the expression of this Ly6C2, which is how we kind of came up with their Ly6C low and Ly6C high expression profiles here. And this has been described a little bit in the literature before about Ly6C kind of differentiating between different monocyte populations, but it was interesting that we were able to find them in the context of spinal cord injury here. So there are more subtle differences, I think, between these two populations as we go on. They share a lot of differentially expressed genes, which is what we would expect, but there are a few that make them unique. The other populations I wanted to mention were the macrophage A and B, because they made up the majority of the mature macrophage population. The transcriptional differences here were much more subtle than what we were expecting. They share a lot of differentially expressed genes, but there are a few that do differentiate them. So one of the most differentially expressed genes is this HMOX1, and then some of the SPP1 as well. And then I think one of the big ones that's very interesting is also a little bit of the ApoE population. So I'll explain kind of why this is important later on, but we didn't actually find major differences between the mature macrophage populations, which have actually been highlighted more dramatically in the literature previously. But the resident or microglia populations did have some pretty distinct differences. So the homeostatic population expressed many of the canonical homeostatic genes that are described in the literature before. So like P2Y12 and TMM119. And then the DAM populations here had a very interesting dynamic. So I wanted to highlight a few things here. The DAM B and C population actually share a lot of differential genes with the homeostatic population. So if you look up where some of the homeostatic differentially expressed genes are, they're actually also expressed by DAM B and C, which we thought was very interesting, and I'll kind of explain why or what we think may be happening here in a minute. But something also that I think is worth highlighting is this DAM-D population, which is present mostly at the 7-day time point, actually also shares a lot of genes with the dividing microglia population. So I'll kind of explain then what we think is happening based on the temporal progression that we were able to provide, and then the differential gene expression profiles here. So we kind of came up with a model of progression, what we think is happening throughout the injury time point, and how these subpopulations might be contributing to each individual time point. So I'll start with the microglia here. Obviously, in the injury condition, the majority of them are in the homeostatic state. But after injury, they go into the disease-associated microglia program, starting with the DAM-A here. And then based on the differential gene expression, we think potentially the DAM-A cells will split maybe into two different pathways of differentiation. So one of them will go into a state of dividing, which then leads onto the further DAM-D population here. So as I mentioned, the DAM-D shares a lot of the differential expressed genes with the dividing population. So this is a potential logical progression. And then the other progression is the DAM-B and C population, which share a lot of differentially expressed genes with the homeostatic state. So we think that maybe some of the disease-associated microglia will continue to go down this disease-associated path, and then some of them begin reverting back to a homeostatic state. So after we get into the DAM-B and C progression, they might start to go back into this homeostatic program. And again, this is all speculation based on the transcriptional profiles that we are provided. So then, this is for the microglia, looking at the peripheral myeloid populations, specifically the macrophage population, so the major monocyte to macrophage transition here. Let me move this over. So obviously in an uninjured condition, the blood-brain barrier is intact, there's no vascular disruption, so there really are no monocytes or macrophages within the central nervous system. But as soon as the injury occurs, the blood-brain barrier is disrupted, these monocytes are then able to come into the injury site and begin their differentiation. So we have two different models of what we think could potentially be happening based on, again, temporal progression and differentially expressed gene profiles. So the first one here is that the monocytes come in from the vasculature in this Ly6C high state. They then will transition into a Ly6C low state. So the downregulation of Ly6C gene is, we think, is a hallmark of the start of differentiation into the macrophage state. So once Ly6C is downregulated, it then transitions into the macrophage A and then macrophage B. Something that could also be happening is that these two monocyte populations are independent of each other. So rather than a differentiation from one state to the next, they come in both as one is Ly6C high and one is Ly6C low. And then based on that, based on the shared differentially expressed genes, the Ly6C high population would go into the macrophage B mature state, and then Ly6E low would go into the macrophage A as time progresses. So again, this is just based on the transcriptional profiles that we had, but this is something that we're interested in confirming further down the line. So in addition to just looking at the single cell data, we wanted to start confirming some of this at the protein level. So we wanted to start maybe validating some of this using flow cytometry. So there's a lot of populations to pick from. So we started with some from the resident microglia and then some from the peripheral macrophage populations. So we wanted to first look at the homeostatic to disease-associated transition to see if that really does exist. So the homeostatic to DAM-A progression, and then looking at the major macrophage populations present here, so A and B. So a little bit of methods on how we did this, very similarly to the single cell transcriptomics. We did a 65 kilodyne spinal cord injury, an 8 millimeter section was taken, and then processed for flow cytometry. And I will go over the markers we used to differentiate these in a minute. We only did uninjured 1 and 7 days here, mostly because of the processing it takes for the flow cytometry. There are a lot of samples to handle at one time, and if you handle too much, it actually compromises the data after a while. So we focused on the uninjured to 1 transition, and then the 1 to 7 transition. So moving forward, starting with the macrophage populations here. This is the dot plot that I showed earlier. Some of the marker genes that were used to differentiate these populations. So we decided to use the CD63 to differentiate the monocyte population from the macrophage. So not necessarily between the Ly6E high and low, but just between the monocyte versus macrophage state. So you can see here CD63 is expressed by macrophage A and B, but really is not highly expressed by the monocytes. So we chose to use this marker, and then we chose to use ApoE as a marker to differentiate between macrophage A and B. So if you look here, the difference is pretty striking. So this is an example of what we saw in flow cytometry. So we first started with each individual time point here, uninjured 1 and 7, and then on the axes here are the labeled markers that we use to work out the gating. So we plotted CD11B against CD63, and over time you can see the progression of the monocyte population change. So as we would expect, there are very little to no cell positive cells in the uninjured condition, but over time the monocyte population increases and then decreases. So this is a way for us to pull out the monocyte population and then just look at the macrophage A and B state. So once we're able to do this, we then come down to the ApoE marker to differentiate between the macrophage A and B. So we plotted this against CD11B, and you can see over time, at 1 day, we have the macrophage A population that then goes in to the macrophage B. So it is a more subtle difference, I think, than what is seen transcriptionally, but it's definitely a difference in proportion here. So we could quantify this to see if this was actually proportionally different. So here, as I said before, in the uninjured condition, there are really no cells, so there's nothing to quantify. So we plotted the 1 and 7 day here. And you can see the monocyte population really dominates more than 50% of all of the peripheral myeloid cells here. There are some macrophage A and B populations starting to show up, but the monocytes really make up the majority here. And then by 7 days, we still do have CD63 negative monocytes, but the majority of the population are mature macrophages. So as we would expect, macrophage B makes up the majority of the mature macrophages here, and then macrophage A is significantly less. So this was very exciting that we were able to temporally confirm the progression of these populations over time. And now moving into the microglia population, as I mentioned, we were looking at the homeostatic to disease-associated transition, so the H microglia to the DAM-A. So in order to identify these populations, we used P2Rate12 as a marker for the homeostatic population, which then drastically decreases in DAM-A, and then also the MSR1, which is a scavenger receptor expressed by these the DAM-A population, which is virtually non-existent, as you can see in the homeostatic population. So these are the two markers we chose to identify these versus in flow. So here, as you can see, the P2Y12 is plotted here, and then the MSR1 marker is plotted here. So in the energy condition, as we would expect, the majority of the are in the homeostatic state, which is shown here by their high expression of P2Y12. There is a small expression here of a population that is expressing P2Y12 to a lower degree, but there is no MSR1 positive signal here. Now, very interestingly, when we get to 1 day, we do have a lot of MSR1 positive signal, but it's also high in P2Y12, which is not something that we were expecting. But by 7 days, we do see a decrease in the P2Y12 high MSR1 cells, and then the majority of them are actually down here. And then also interestingly, we have a population here that is low in P2Y12 and MSR1. So maybe this will help a little bit. This is the quantification and how we're kind of determining how these cells changed over time. So as we would expect, the homeostatic population really dominates the uninjured condition. And then over time, the disease-associated profiles come into view. So we did mark this population up here, transitioning DAM-A, which is not something that we picked up in our single cell transcriptomic analysis. We think that this may be due to a change in, or a discrepancy in the RNA to protein translational profile. So by 1 day, all of the P2Y12 protein might not be, it might stop being translated, but it's not, it hasn't actually been cleared from the system yet. So we think that there's a lag in what the single cell data says, and then what the temporally what the protein profile would be. But there is still the progression of the disease-associated state from the DAM-A into what we're terming as the rest of the DAM. So as you can see, the B and C start to kind of come into view more over time. And then this P2Y12 low, MSR1 low population, which is really more of what the DMD population is down here. So they really don't express either of these markers. This comes into view at 7 days as well, which is consistent with what we saw in our single cell analysis. So we do have some more validation to do. I'm working on validating the DMD population with this IGF-1 marker here. But so this is kind of a preliminary estimation of how the rest of the DAMs are also showing up over time. Okay, so now after looking at the transcriptional heterogeneity within this population, I wanted to start to understand how they're changing other pathologies over time, or how they might be interacting with the other cells involved in the pathology and influencing its progression. So there are two pathologies that I want to talk about here. These hypotheses came from some data that a previous student in our lab, Yunjiao Zhu, found. So he was also very interested in how myeloid cells interact in the immune system, in the spinal cord. So he used a macrophage depletion model in order to look at the injury site in response to a depletion of macrophages. So some of these hypotheses came from the data that he's also found. So we're going to look first at the revascularization event that occurs in the injury site, and how the myeloid cells might be influencing the progression of that event. So here is the data that kind of gave us this idea. So this is from Yoon Zhao's paper back in 2015, where he used a drug called clodronate to deplete the peripheral macrophage, the peripheral myeloid population. So the macrophages were depleted in the injury site, and then he looked at various pathologies to see how that would change. And one of the things that he found was that the revascularization of the injury site is drastically decreased when the macrophages are depleted. And he kind of left it at that. So I took this and was interested to see if there was maybe a mechanism for the macrophages to be interacting with the cells that revascularize the injury site, and if those two pathologies can be interacting. So this is the first hypothesis here, was that the myeloid cells, not just macrophages, but any myeloid cell could be contacting the vascular associated cells and influencing the revascularization. So in order to do this, I wanted to get a better idea of the subpopulations that make up the vasculature. So I took the endothelial cell populations. Actually, James actually did this bioinformatically. He isolated, similar to how we did the reclustering analysis for the immune cells, did the same thing for the vascular associated cells. So the fibroblasts, pericytes, and endothelial populations were isolated and reclustered to look more intensely at maybe some of the subpopulations that could be influencing this revascularization event. So we found something very interesting. This is independent of the immune component here. This is another UMAP of the vascular cells. And something very interesting that we found was a population here that we were able to annotate as a tip cell population based on canonical genes from the literature. So if you're unfamiliar with how this works, this is not just in the CNS, this is in many other tissues in the periphery as well. In response to a disruption of the vasculature, certain molecular cues in the environment will cause endothelial cells in the existing vasculature to go into a tip cell state where they butt off from the intact vasculature and start to form new branches and basically revascularize the tissue that was wounded. So this has been extensively characterized in other tissues, and the major signaling pathway that we know that is kind of involved here is the VEGF receptor, and VEGF receptor signaling pathway. So the VEGF ligands here, A, B, C, and D, and then the VEGF receptors are also listed down here. So this is a simple schematic of how tip cell interaction is thought to happen in the periphery. So we wanted to see whether maybe this was recapitulated in the spinal cord injury site, and if maybe the immune population could be a source of these VEGF ligands that could be causing the endothelial cells to go into the tip cell state and start revascularizing the injury site. So before I show you the data, I want to kind of go into how we started analyzing these interactions between the immune cell population and other cells. So we wanted a quantitative way to analyze interaction. So we looked into the literature, and after a lot of searching, we found a method that we modified. It's called CellPhoneDB, and it's a very simple algorithm that uses the expression of any given ligand on any given cell, and then its corresponding receptor in any other given cell that you're interested in looking at interacting. And it's basically an average of that. So this is the kind of the equation of how this interaction score is calculated, but it's just the log values of the ligand and receptor that are known to pair together. And it's an average of that. So it's more of a crude way of analyzing interaction, but we wanted an idea of just whether a ligand and receptor are present on these cells to even see if interaction is possible. So this is, for example, so the cell A would be a microglia or a macrophage, and then the cell B would be an endothelial cell. And then we would be looking at the interaction score between these two, and the score would be an output of the relative expression of a ligand on the first cell and the receptor on the next cell. So this is kind of how we moved forward in looking at interaction between all the different cells we were interested in. And when we look at this is a dot plot representation of the interaction scores. So I know it's a lot, so I'm going to kind of orient everybody on how to read this. On the top y-axis, the top x-axis up here, these are the cell populations that are providing the ligand. So these are the immune populations that we were interested in, and then we also included some of the existing vascular and glial populations that we found scores for. So the ligand expressing cell is up here. The ligand receptor pair of interest is on the left. So for example, PLGF is the ligand and NRP1 is the receptor. So this is any given annotated by the score. And on the bottom axis here, we just looked at endothelial cells. So a common method that is described in the literature is that existing endothelial cells in the vasculature will bud off from the intact vasculature and turn into these tip cells. So we wanted to really see how this VEGF receptor signaling axis was being influenced on the endothelial cells themselves. So we plotted this over time with various ligand providing cell populations given up here. And then basically the strength of the score is indicated by the color. So a higher score here is basically inferred as a higher expression of either the ligand receptor. So as we can see, and there's not really a lot of interaction here in the energy condition. This PLGF is kind of expressed a lot of the time. So we think maybe this is more of just a vascular homeostatic idea where it's just the turnover of the vascular cells. But then when we get into the injury state here, we have a lot more of the VEGF ligand receptors being expressed, meaning that access can actually start interacting. So something that was exciting to us was that this VEGF-A and VEGF receptor 1, which is it's a canonical pathway used in tip cell specification in the literature, is actually present in this monocyte population that shows up at 1 day. So there are other cells that participate in this. So we weren't hypothesizing that myeloid cells are the only cells that are able to induce this revascularization event. So there are the existing fibroblast population, and then actually the astrocytes are also showing some interaction here. We can see over time, even then, as the monocytes go away and the macrophages start to become mature, the interaction score kind of persists. So this kind of gave us an idea that's one, this axis is active, and then two, the monocyte population that comes in very early after injury is contributing to this axis working, which is something that means maybe the tip cells are actually showing up. So we wanted to kind of confirm this, that the tip cell population is actually showing up after injury. So what we used in order to do this, we did in situ hybridization and immunohistochemistry mixed together in order to identify tip cell mRNA present in the endothelial cell population over time. So this is the uninjured condition here, and these cells are kind of, to orient everybody here, the box here is where these bigger images are taken. So we have the GFAP to mark astrocytes. So we use this to mark the injury site so we can see how close to the epicenter we are imaging. And then this photocalyxin is a marker of endothelial cell populations. And then we used the marker apelin to mark the tip cell population. This is very well described in literature as a marker of tip cells. And this is the merged image. So you can see there is a little bit of apelin staining randomly in the in the tissue in the uninjured condition. There are some papers that suggest that neurons can express apelin in the uninjured state. So we think maybe that's where these apelin positive staining is. And then as we look right after injury, where we would expect this VEGF axis to be active, we get a very different picture. So as we look over time, we can see these long strands of endothelial cell populations. And then inside those populations, we have a very drastic increase in the apelin positive staining. So the apelin is done by in situ hybridization. So each individual dot is corresponding to an mRNA transcript. So we can see, it's visually striking the difference between the staining here, that these endothelial cells are now very highly expressing these apelin positive populations. And then as the VEGF axis decreases over time from the macrophage population, we see sort of a decrease in this stark expression. So it's definitely still there, but it does decrease over time. So this was very exciting, not only to identify a tip cell population in the spinal cord, but to identify the peripheral myeloid population as a potential source of priming these tip cells to go into the vasculature and start revascularizing the injury site. So I know I'm running a little short on time, but I don't have too much left. So I want to get into the next pathology here, which was scar forming. So both the glial scar that forms around the injury epicenter, and then the fibrotic scar that will form in the center of the lesion epicenter. So going back to Yun Zhao's work, something else that he found very striking from his macrophage depletion studies was that in the condition of the macrophage depletion, the fibrotic scar that forms in the center of the lesion here, as you would see, is also drastically depleted when the macrophage population is taken away. So this was very exciting for our lab, and then I've been very interested in this over time. So I thought maybe there's a potential interaction between these two cells, that the macrophages are somehow interacting with the fibroblast population to create this fibrotic scar in the lesion. So going back to the interaction, I wanted to see how could these myeloid cells be interacting with the cells that form the glial and fibrotic scars. So I went into the literature, and this has been studied by several other labs as well, but something that we're interested in is the STAT3 pathway. So there's a lot of papers on phosphorylation of STAT3 preceding astrogliosis after spinal cord injury, and then fibrosis preceding a lot of other peripheral fibrotic events, such as in the lung and the liver and things like that. So it's a highly supported idea, but we just haven't been able to really test it too much in our own hands yet. There are previous students that have looked at somehow a little bit of how the OPC population might be looking at contributing to the STAT3 activation, but I was more interested in how the myeloid cells could be potentially activating the STAT3 population, or excuse me, the STAT3 pathway in the astrocytes that form the glial scar, and then the fibroblasts that form the fibrotic scar. So just very briefly, the IL-6 family members are kind of some of the most canonically characterized activators of the STAT3 pathway in various cell types. So there's a lot of them, and I'm not going to go through each one, but basically this GP130 receptor will dimerize with one of the co-receptors, depending on what ligand is active. So for example, if LIF is binding to the GP130, it needs to dimerize with the LIF receptor, and then the STAT3 activation cascade will be activated. So this is kind of a mechanism that we were interested in looking at as a potential way that these immune cells could be interacting. So we went back to the interaction score that I talked about before and looked at the IL-6 family members from myeloid cells interacting with the IL-6 family member receptors on these scar forming cells. So this is kind of what we came up with. So it's a lot of information, but I just want to point you to kind of what we found that was important. You can see on the left here is as similar to before, the ligand receptor pair that's given. So for example, oncostatin M is a ligand and oncostatin M receptor is the receptor for that given pair. The cells that are expressing the ligand are shown on the top, and then the cells that are expressing the receptors are shown on the bottom. So this is characterized at each individual time point here. So something that we found that was very interesting, this axis really isn't active basically at all in the uninjured condition, and then after injury, we see an increase in the scores here. So something that we thought was very interesting, one, we see a very high score here from lights, both the Ly60 high and low, more of the Ly60 low monocytes here, and then a little bit for the DAM-A population from the microglia. But really the only cells that gave us any, the only ligand receptor pairs that gave us scores were this oncostatin M and the oncostatin M receptor. So as I mentioned before, these receptors have to dimerize with the GP130 receptor, which is termed IL-6ST for its gene name. So basically what we found was that right after injury, this oncostatin M and oncostatin M receptor with the GP130 that's necessary for dimerization have high scores with the monocyte population, and then a little bit of the disease-associated microglia A. And then over time, this starts to kind of go down. The scores start to go down a little bit, but they are still showing positive scores over time. And if we look at what cells are actually expressing the receptors, there you can see the astrocyte population here has a high score with the monocytes, and also the fibroblast population, which make up the fibrotic scar. So this was exciting because we found one specific ligand receptor pair that we think may be able to activate STAT3 in these scar forming cells, potentiating the scarring response. So we wanted to see whether this access was actually active in vivo. So the first step here is to actually stain for some of the protein, and then look to see if this is actually present in our tissue. So again, we went back to the in situ hybridization immunohistochemistry method. So we first looked at the oncostatin ligand here. So just to orient everybody, we have our GFAP to mark the lesion site. We use CD11B as a marker for myeloid cells, and then this is the oncostatin M. So the CD11B and oncostatin M are in situ, which is why they appear as dots here, mRNA transcripts, and then the GFAP is done by immunohistochemistry. So to kind of get an idea of expression over time, we came up with a method of quantifying the number of these OSM positive dots or transcripts over time. So basically what we did was we took an individual DAPI positive cell here, anyone that was positive for CD11B mRNA, we counted the oncostatin them there, and then anything that was negative for CD11B, we also counted the oncostatin M, and then looked basically at the expression between the CD11B positive or myeloid population and the CD11B negative or non-myeloid population. So as you can see here, in the uninjured condition, there's really not a lot of oncostatin M transcript expressed, but very soon after injury, at one day, the CD11B population or myeloid population starts to express the OSM transcript, but there is no change in the CD11B negative population. And this transcript expression actually decreases over time, which is consistent with what we saw in the interaction score plot So this also tells us that the myeloid population is the only source, or likely the only major source of oncostatin M ligand. So it's likely the major activator of this STAT3 cascade. So of course we can't have an interaction without the receptor. So we looked further, and this is all immunohistochemistry. So we used PDGF receptor beta to mark the fibroblast population and the oncostatin receptor to bind with the oncostatin M ligand. So this is kind of this yellow line outlines where the injury site would be. So out here is the kind of the glial scar area, and in here is more of the fibrotic scar area. And we used a signal colocalization algorithm to look at the overlap between GFAP and OSMR, and then PDGF receptor beta and OSMR to see to infer expression. So basically, with the colocalization between the fibroblast population and the oncostatin M receptor, this signal drastically increases over time. So the oncostatin receptor is being expressed more highly as the injury progresses, and this trend is actually recapitulated in the astrocyte population as well, which is consistent with our single cell data. So this at least tells us that the components required for this STAT3 activation in these cells are present, and that the myeloid population is the major, if only, provider of this activating ligand. So that's where I will leave you now. I'm working more on a functional study here to actually test this in a knockout situation, but all of the components required for interaction are actually there. So very quick summary. The first part of this was really looking at the heterogeneity transcriptionally between the myeloid populations. So we were able to identify several subpopulations of myeloid cells, both from the periphery and within the CNS, that we think might be differentially contributing to the pathological progression. Looking at how it might be interacting with other different cell types and other pathologies. We think maybe the VEGF signaling axis is active between the immune cell population and the endothelial cell or TIP cell population, maybe priming or initiating this revascularization event after injury. And in addition, the scarring populations here as well, so the glial and fibrotic scar populations, we think are being activated via STAT3 using this oncostatin receptor signaling access that's actually being provided by some of the myeloid populations as well. So not only are the myeloid cells after injury very diverse transcriptionally, but they also exert a lot of influence on the progression of other pathologies as well. So I think one of the major takeaways, at least for me, was that using any type of intervention in the immune cell population will likely affect the progression of other pathologies within the spinal cord as well. So they're all very, very dynamically connected. So with that, I will end and thank many, many people for this help. My mentor, Jay Lee, has been extremely helpful and supportive in the entire process. This was very different for our lab, kind of jumping off from our classical histology background. So we took a leap with this, but I think it really, really paid off. James Cho has done all of the computation. This would have been absolutely impossible without him. So thank you very much, James. And the rest of our lab, who have been very supportive and very helpful. And Pantelis has been wonderful with troubleshooting ideas and figuring out really the scope of how we want the paper to come out, and what we really want to focus on, what we think is important. And then my dissertation committee for always being very supportive and guiding me in the right way. So thank you very much. And I think I can take questions on here. I'm not sure. Everybody seems to be muted, but.
If they have any questions, please unmute yourself and yes, ask a question.
Okay.
Any questions? Any questions?
Quite all right.
If you don't want to ask the question, you can always put it in the chat and we can read it for you.
You can email me too if you're interested in doing single cell, or if you have any questions about the data. I'm happy to talk about it at any time, not just now.
Hey, Lindsay, this is Colleen. Can you hear me?
Loud and clear, doctor.
What an awesome talk. Just beautiful data. It's very interesting. I was curious, did any of the disease-associated microglia subpopulations align with an M1 phenotype? Did you see any with an M2 phenotype?
So we did kind of look, we went back into the literature, looked at kind of the canonical markers that are supposed to define these populations. They express maybe one or two genes here, but they don't align with the profile. So M1 and M2 really have a distinct transcriptional profile that is described in the literature, and they really aren't aligning with the profile. We would expect maybe a gene here or two to be expressed because they're used in the inflammatory process, but that was something very striking that we found, was that the subpopulations don't align with the M1, M2 kind of dichotomy that's described already. Thank you.